All right, I'm pausing the recording. I hope you're all fine with being recorded and that being able to be viewed later on by others. Coolio. First of all, welcome, obviously. Super happy to have some people here, even though I was not expecting to have 4 people out of 20 on the mailing list to attend the first meeting. I expected more of a drop-off, but it is what it is, as Trump would say. And so you may remember from the invitation email, and I think that put a lot of people off, that I set this to be 3-hour-long meetings every week. And while I would like for everyone, if possible, to reserve that kind of time slot, I don't really expect any of these meetings to run for a full 3 hours. So the way I envision these meetings to be run, and I'll gladly take charge of most of this, is that a volunteer, mostly me, because I'm interested in this a lot, will go through the lecture material on YouTube by Richard McElraith and the book Statistical Rethinking by the same guy, and compile a sort of summary of it. That can be a PowerPoint presentation. I prefer to do it that way because it gives me something to talk about and to flip back and forth to, but it doesn't have to be. And if someone wants to present a certain session and they don't feel as though they have grasped the full concept of what they should present, that's not an issue. This is not about you being an expert coming in here and then we circle jerk about it, how much of an expert we already are. This is us not only admitting to ourselves, that's why we're here, we want to study some more, but also admitting to the others in the group that we do have some need or wishes to study Bayesian analyses a lot more. And for that, it's totally fine to admit when someone doesn't understand something. Sam and I have been in a similar study setup with the University of Oxford this semester. And I'd like to just repeat what was said there, which is that if you feel as though you have a stupid question, you can be sure that probably 3/4 of the rest of the group have the exact same question and just aren't bold enough to speak up about it. And I would like us to have a kind of atmosphere in here where we can just pipe up with questions whenever we feel as though we have some more need for clarification. That being said, I don't know if you guys have had the time, energy, or mood to go into the preparation for today's material yet. If you don't, I'm going to quickly go through it, and it's totally fine if you do not. Please show up even if you haven't managed to get through all of the material, because you're still going to get something out of it. I will also, by the end of this, upload the PDF of the Statistical Rethinking book and the PDF of some other books that I will be using for preparation to a drive. This is where I will upload the schedule, and this is also where I will upload the recordings, and in another email after the fact of today's meeting, I will send around a link to that drive so that everyone can always stay up to date with what we've got in terms of material. This is also where I'll upload all of my PowerPoint slides for each individual session. And if someone else wants to run a session or I can't be present at a certain session, and then I would be really happy if someone else took over. If it's any one of you guys or any one of the ones watching the recording later on, it would be super cool if there are presentation slides, if they could just be sent to me so I can upload them to the drive. I'm only going to make it read available to people, because otherwise someone might accidentally hit a delete button and then we're all out of some nice preparation stuff. With that out of the way, since we've got a lot of people who don't know each other in here yet, how about we just go through and introduce ourselves right quick? I can gladly lead the charge on this one. Name's Eric. Hi. I am a relatively fresh PhD student, so I'm just about to close out my first year at the University of Aarhus, so I've got 2 more after this. I have so far been working a lot with remote sensing data, and through that have gone into some more climate reanalysis data sets with a climate scientist pal of mine, for which I've written an R package to do some downloading of the climate reanalysis and statistical downscaling to reach higher spatial resolutions. And in doing so, I realized just how much uncertainty there is associated with our climate data sets that we usually use for macroecological experiments or analyses. And it is because of that that I've been thinking, how can we include that explicitly into our analyses in macroecology? And because I'm currently trying to combine those high resolution climate data with interaction networks of species, I was looking for some sort of statistical method that can do really nice error or uncertainty propagation from one step and one data set to the next, and also identify some real causal pathways, which landed me on graphical methods, both of which funnel me into Bayesian networks. And I have already read a bit about that in the McElreath book, so I'm really looking forward to getting into this. So that's where I'm coming from and what I want to get out of it. I'm just going to go through by order on my screen, if that's fine with you. And Gregor, if you could just tell us a tiny bit about you.
Yeah, of course. Hi, my name is Gregor. I just finished my first year PhD in Bayreuth with Manuel Steinbauer. I studied geology. I'm a paleobiologist, so mostly working with fossil data, large fossil databases. So I'm not going out in the field and collect anything. And to be like in detail, I'm a conservation paleobiologist. So I try to use the knowledge we get from the past and apply this to the recent climate change debate and crisis. So while doing so, I've noticed, probably just as you, Eric, that there's a problem, and this problem with paleobiology and ecology is that we do quite a lot of cool stuff in paleobiology, but people in ecology don't see that, and they don't include that in their research because we're working on different scales. And so I was trying to find a method to implement the knowledge and the facts we get from the fossil record into recent neontologist research. And I think Bayesian statistic could be used for this, where you basically just use the fossil record or the knowledge from the fossil record as a prior. So this is how I started to get into Bayesian analysis. I've already started to read the book like a year ago, but came to the chapter 4 and then like life happened, and now I'm here. Looking forward to the next weeks.
Well, glad to have you, Gregor. Thanks for that introduction. Sam, you're up next.
Hi guys, I'm Sam. I've just started my PhD at Oxford. I'm really a life history guy. My data is nowhere near as cool as your guys' data, because I really work with either simulations I build or with Drosophila, my lab stuff. I came here because I think maybe 3 weeks ago, Eric and I had a nerd out session with another guy in the States, and he just brought up the idea of Bayesian networks. I said, those are cool. Hi, I'm starting a thing in a few weeks' time. And it seems really cool. And I think I could use it actually with some of my demographic buffering stuff, because currently with those data, no one really knows how to analyze it. Use this there. And if not, I'm actually just excited to learn about Bayesian statistics as a thing. And if I can use it, all the better. So that's where I'm at.
Thanks. Just calling back to that nerd out session, I am still 100% certain that you'll find Bayesian analysis really useful for what you're doing. Anna, you're next.
So hi, I'm Anna. I'm also from Bayreuth. I work there in the Department of Biogeography, and my research is focused on islands, on different aspects, mainly on the anthropogenic impact on islands, ranging from restoration ecology, really going into the field collecting data, to invasion ecology, I'd say. And two of my main focal islands are the Galapagos Islands and the Canary Islands. And I'm still looking at how I can implement and use Bayesian statistics for my PhD and my research.
Cool. I am already, and this may seem like something everyone says at the beginning of this course when everyone's introduced themselves, but I am legitimately fascinated by how different our backgrounds are, just with the 4 of us already. And definitely once the others start trickling in on the next few sessions, looking at all of the people in the recording right now. I I think we will have some really neat discussions of what people want to do with Bayesian analyses and with others weighing in of how they think it can be done. That being said, I am going to go straight into sharing my screen and walking you through what the lecture for this week on YouTube and the chapter in the book had to say for today, and what I took away from it. So this isn't trying to be a full summary of what we were talking about or what the book was trying to tell us. This is more of a what I took away from it. And if anyone else took more away from this or thinks that something is unclear of what I'm about to say here, feel free to interrupt me at any given point in time. I'd much rather have a lively discussion right away than having this be a lecture style interaction where you have to write your questions down before we finally get to answering them. That being said, I'm not going to drone on for 90 minutes. I'm just going to go into this, and I think we'll be done in just a flash here. So the title, and I think the storytelling was quite good in the book so far. I actually did out loud say to myself, I wish I could write as well as Richard did in this book, because I felt as though it was quite compelling to read this. He talked of the Golem of Prague story, where he then compared the Golem story to us using statistical methods which are unthinking themselves and function as automatons or robots. So getting into it, the first thing that really stuck out to me, which came up in the lecture material, was that he said that our methods are designed for settings in which we have randomization and an experimental control in a system in which the effects are large. And while all of this rings very true for what we have experienced in introductory statistics courses and in probably the way in which we think about our analyses for the most part, at least for myself, that's true, unfortunately, ecological analyses usually do not meet these expectations. And despite this being quite obvious to people once they're told about it, we still have these kinds of flowcharts. And here I have to say I'm quite guilty of doing this myself, of not only selecting some tests and methods every now and then using a flowchart, but most importantly, teaching it that way. And like it was stated in the book, and I fully agree with this because it gives me a bit of absolution on my end, this is not a bad thing to do necessarily. For people who will use these kinds of tests in very well-controlled settings with large effects and randomizations, so mostly experimental setups where this is actually possible, this is absolutely sufficient. However, for people who really go into large ecological questions, such as the four of us right now in this chat, we have to recognize that those are tools instead of logical or rational entities. So these are inherently, and this is where the Golem comes in, unthinking, as they do not test for whether certain assumptions are met. And in fact, they do not end up telling us the ultimate truth about our data because of hypotheses sometimes pointing to multiple different models, and a whole nother can of worms opening up when we talk about null hypothesis significance testing, NHST, which has been heavily criticized for a long time and is probably a good discussion point for later on when we get started to discuss a bit just today what the preparation was on. So the big challenge that arises out of here is that whenever we apply a statistical method to a research setting, we do need a lot of knowledge on not only our research setting itself, but also the statistical method, because we need to understand how it works instead of using it just like the automaton it is, because then we may run into faulty predictions, faulty explanations, which could have been avoided if only we understood better what an actual method is doing. And this comes back to a quote by a guy whose name I unfortunately forgot, but is one of the statisticians for the US Cancer Institute, or at least was when I when I saw the quotation of it. And he said to treat statistics not as a cookbook where you follow a recipe, as you can see on the left-hand side, but really treat statistics as its own science. So devote more time to understanding how you do certain statistics. This then leads us into a lot of confusion of hypotheses and models and hypothesis inference testing. And just taking a few points from the book here, I really like the short revision that McElraith put into this first chapter of revisiting what is a hypothesis, what are process models, and what are statistical models. Because before reading this, I had honestly never heard of someone calling something a process model. I like that he did it, and I'm probably going to do that from now on myself, but I never heard anyone refer to the concept as such. So just as a takeaway from this, for me, hypotheses are usually vague and verbal, and they are too vague to actually test in a statistical sense of the term. With those, we can then build process models, which are expressions of an expected causal structure that give us some kind of understanding of a cause-effect relationship. However, without data, they are not mathematically testable, and even if we have data, testing them will be very difficult. So in turn, process models can usually be taken as the base to engineer statistical models from, which we can then use to test associations, but not necessarily causal relationships, depending on how we engineered them. And I think this is an important step that often gets skipped, and I have skipped myself quite a few times, where we incorrectly equate the hypotheses that we have with statistical models, even though there's another layer in between the two, and there is seldomly a clear line to be drawn from one hypothesis to one specific statistical model. As in, you can test one hypothesis with multiple models, and you can test multiple hypotheses with just one model. And probably neither of these models that you will run have the ultimate truth about the hypothesis in question. And while this may sound like McElraith, and by extension me right now, is trying to refute any kind of statistical work that we have done so far, just by virtue of saying, well, it's not the ultimate truth, so why do we bother? I would like to believe that is something to be worth of embracing. We know that we do need multiple models to tell a full story or show the whole picture of a certain research setting and research question. So instead of saying this and that model should have been run, therefore everything else is invalid, I at least would argue we should run an ensemble of models, just like it's done in climate reanalysis studies, which is where my background lies currently, to then come to a consensus of where we arrive with our final conclusion. So the challenge, again, that is posed here is that hypotheses do not imply unique models, and models, on the other hand, do not imply unique hypotheses. The goal to get out of this is to engineer methods that address specific needs and hypotheses themselves. So not to just go through a flowchart and pick out whatever is suggested by a given flowchart, but to really go into the methods themselves and see where you can pick and choose certain aspects from and put them into one method yourself. This then leads us into how we have been taught about statistics so far. And there's this whole debate between frequentist statistics and Bayesian statistics. I believe we have all heard about this. I'm still going to give a tiny summary of this as well, where I also delved into another book for this. A lot of Bayesian statisticians say that Bayesian is a lot better than frequentist statistics. Frequentist statisticians, which is what we are taught at undergrad and mostly grad courses, will say that frequentism is so much more precise than Bayesian analyses and so much easier to do, so that's why we should stick with that. I, again, like that McElraith didn't necessarily take sides on this argument. The other book I went into did take sides heavily in favor of Bayesian statistics, which should come quite natural in a book all about Bayesian statistics, but I still like that McElraith didn't just outright say that one method is better than the other. However, when we look at how these two methods compare, frequentist statistics compare sample frequencies that we have obtained from always just a limited sample, because we can never sample everything of one quantity in the entire world, to an imaginary repeat sample frequency, which means that this is only applicable in large samples. Most frequentist methods work on the basis of maximum likelihood estimates, which are only unbiased asymptotically, meaning only for infinitely large data sets would we have completely unbiased MLE, maximum likelihood estimates, which right now already shows us how with ecological data sets, especially if we don't have especially big ones, which will probably be an issue for Sam, at least, with his probably somewhat limited data sets that come out of the experiment. Not trying to down talk what you're doing, Sam, by the way, just trying to highlight the huge sampling effort that you will have to go through in your PhD. Just wanting to be diplomatic here, because I have huge respect for what you're about to do sampling effort wise, and I could never. That being said, this is probably where frequentist statistics will run into some issues. Whereas on the other hand, Bayesian statistics only test or compare data to assumptions that a certain hypothesis or process model makes, meaning we check how true our hypothesis is given the data itself, and not the other way around, which is a really nice way of thinking about how we view the world, I would like to think. This also means that this is applicable for any sample size, because the calculations are done very differently from frequentist statistics, and we'll get into that. I had a small glimpse of the next few chapters, probably over the next few chapters. So right now, probably best not to go into too much depth on this for now. But what I think is very important here is how randomness is viewed between the two different methods, or two different paradigms. And one is that with frequentist statistics, randomness is usually assigned to be a part of nature. This is why we have those distributions that we compare things to another. These distributions have that random aspect baked into them. Whereas on the other hand, and I think that makes much more sense for most of us ecologically minded people, randomness is part of the data. It's down to sampling bias, sampling efforts, sampling methods, sampling regime. But in nature, everything has a cause-effect relationship. That is the underlying paradigm shift between the two. And that makes Bayesian analyses so much more compelling to me, because that is how I would like to view the world. Now, another thing that comes in here and usually leads to a ton of confusion, not only in introductory stats courses, which I have taught, and I've seen this done a lot, and I have even fallen into that trap myself a few times when I started out with stats, and I like to believe I'm not doing that as much anymore. Still happens every now and then. Frequentist outputs are often falsely interpreted as their Bayesian counterparts. So for example, the p-value is often identified as the probability of the null hypothesis being true, which isn't the way to interpret the p-value, but you will still often hear people say, well, by adding a few more 100 data points, my p-value decreased, therefore my alternative hypothesis just became more likely. And that's an incorrect interpretation of a p-value. But it is a correct interpretation of a posterior probability in Bayesian analyses. On the other hand, you have confidence interval from frequentist statistics, which are often interpreted as, with a 95% chance, the real effect lies within that interval. And again, that's very persuasive, but it's just not true, because that's not how we interpret confidence intervals. Confidence intervals always go to the extreme of, if we were to sample infinitely, in 95% of those infinite samples, the effect would lie herein. And it might sound as though, yeah, that's just the probability of 95% of the actual effect being in this interval. But mathematicians have told me it's not, and I trust them. And I have had a hard time wrapping my head around this. And if someone has a good explanation to this, please do let me know, because I'm still slightly struggling with the differentiation between the two. However, Bayesian statistics give us credible intervals, And credible intervals have exactly that interpretation with them, with a 95% probability in a 95% credible interval, we find the actual effect in this range. So in conclusion, or the way out of this challenge to my mind, is that Bayesian interpretations are often more intuitive to many people, myself included. And for that reason alone, I think it's worthwhile investing the time that we've set aside for these sessions so far to actually look into Bayesian statistics and try and understand more what's happening in them. Now, the next point I want to highlight, and this is a quote from the chapter in the book that I read just today before starting this, was that successful prediction does not necessarily require correct causal identification. And I think that is put so well, and just another way of saying correlation is not causation. Because in fact, everything that we do in frequentist analysis is just another form of a fancy correlation test. And with that, while we may be really good at predicting stuff, we may not be able to identify the correct causal relationships. The book made a super good point about correlating the presence of wind with the rustling of trees. And rustling a tree, not creating wind, but seeing a tree rustle by itself, probably being a really good indicator that it is windy outside. As a matter of fact, that is how we, when we try to gauge the weather before going outside and choosing the right apparel, think about the world. And so models that do not identify the correct causal relationships may be better at making predictions than the models that actually do identify causal relationships. And that's a tricky one, because usually we like to have models that explain as much as possible. And that's how we measure how good a certain model is when we go into model comparisons. However, I would much more prefer models that tell me about how one thing drives another thing, even if it in fact has a higher AIC value, meaning it's a lower performance. And with that, McElreath suggests, and I've seen it on his Twitter quite a few times, where I've been lurking for half a year now, he likes to use directed acyclic graphs, DAGs, which is a way of building networks. And so right up my alleyway with my current project and what I nerded out with Sam a few weeks ago, because they can actually help us identify causal relationships. And I expect the book to go in much more detail on this. But for the uninitiated, just for now, directed acyclic graphs, from my current understanding, and I'm not going to claim it's very good for now, is that directed acyclic graphs are graphs as in networks, where each connection between nodes has a direction. And the acyclic part means that there are no self-loops and there are no loops via multiple different vertices onto a starting vertex, meaning you will never have a loop in your network. So no feedback loops, for example. So last slide of just the summary that I've prepared for today. In the lecture, and you will see that I left out all of the forking data stuff that he was talking about in the YouTube lecture, because that's more of something that will be picked up in the next book chapter. So I thought I'd leave it for that one. In summary of what he said and what the book mentioned, Bayesian data analyses are basically just counting in a special way, meaning that you count all of the ways that the data that you see could have happened given the assumptions that your hypothesis and your process model has made. And then you convert that into probabilities to see how plausible your actual model and hypothesis has been. By the end, you just pick the most plausible hypothesis and run with that one. And that is the one you assume to be as close as possible to true. Not saying this is the one and ultimate truth or the holy grail of whatever you're studying, but recognizing that it could be improved on if you had more data or were considering more models or hypotheses. But this is an inherently iterative process of trying to get better and better of making more and more plausible models. The next step he highlighted quite a lot were multi-level models, meaning models within models where parameters are replaced with models themselves, which then goes back all the way around to what motivated me a lot to start this study group, which is to track uncertainty for each model. Because personally, I've talked to a few people about this, and I know that there may be differing opinions on this. I would much rather prefer a model that gives me even just the slightest of trends as an output by the end, where I can say I have a huge uncertainty associated with it, rather than a model where I find a super significant, in frequentist terms, super significant trend that is super strong in a certain direction that I would have expected, but I can't say anything about the uncertainty. I personally prefer to have some idea of the uncertainty associated with it, because I think it tells a much more, not necessarily clear, but honest story about what the world is like, and more importantly, what our understanding of the world is like. And this, just as a tangent from where I'm coming from with climate reanalyses and statistical downscaling for macroecological applications, I often see biologists, me included, when we get a data set, assume that if there is no uncertainty records in our data set, then the certainty of every data record is 100%. And factually, that just cannot be true. And as a matter of fact, especially when we go to those large scale climate data sets, my biggest gripe currently is with WorldClim. Sam will have heard me ramble on about that quite often, especially downscaled precipitation records, which physically just don't make any sense. Many of these gridded observation products that have been used so far before the advent of climate reanalysis datasets haven't provided the user with uncertainties for every data record, for every cell in the rasters that one can obtain. And so people have just taken these as gospel so far without acknowledging that they are working on gridded observations which have a huge sampling bias in certain regions, and with the interpolation methods used on top, which for some variables just aren't permissible and have never been, but have been done anyways, people have just assumed that what they have been given in terms of data are absolutely 100% correct and certain, which, again, to circle us back to Bayesian analyses, isn't the case. And I would like for people to be able and acknowledge that it's necessary to track the uncertainties of the different data sets throughout their whole process of statistical analyses, just because I think it tells a much more complete and honest story of how we understand the world to be. Lastly, with model comparison, what we have usually done, or what we usually do in frequentist statistics, and I'd like to do away with that for myself by the end of this study group, which will run for quite a while. What we do usually is we try to falsify a null model by doing model comparisons, to then argue, because we have falsified a null model, which we often build in such a way that it has to be worse than what we build on top of it. Because usually even inputting marginally explanatory variables into a model will already be better than no predictors at all. So falsifying a null model is often equated to answering something about the truth of a hypothesis, which shouldn't be the case. In contrast, model comparisons should be done to compare meaningful models, to safeguard against overfitting, and this comes back to the consensus that I was talking about earlier by using a suite of models. Make sure that causal inference is not just drawn from one model. And even if it is, we do it by using the most meaningful model. And in many frequentist approaches, that's already done. We do model selection to identify only the most appropriate parameters, for example, to get to better inference and predictions. However, all of these are still just correlations, so associations between different variables, instead of actual ideas of causal relationships. And I think that really comes back to the big point that I want to get out of this, is how to better identify the causal pathways in nature rather than just the associations. Now, having droned on long enough for this, we've actually reached the end of my summary of this. So I'm just going to stop the screen share. And if you want me to go back to any of the slides, I'll gladly reopen it and get back to it. I just want to see everyone's faces, so I don't feel like I'm talking just to a webcam as much. So this is the way I do these summaries. If you want to do a summary like this yourself, you do not have to do it like this. You may have your own style, and if you don't think what I'm doing is good, please just let me know. And you don't even have to say it in front of everyone else, just shoot me a message. And I'll gladly revisit how I'm doing stuff if it's not valuable to people. But with that out of the way, and if there's something you want to get out right now about this being garbage and me having to do much better.
Eric, it was awesome. Eric, it was awesome. You're all good, mate.
Thanks. But what I want to get into, without fishing for compliments here, is getting to a discussion of what did you take away from the chapter? Was there something that you struggled with in understanding? Or is there something that you think should be added onto it? And the floor is yours.
So for me, I think the most amazing part of the first chapter was probably quite, when you start a book like this about Bayesian statistics, you expect that the book starts with, hey, these are the problems with frequentist analysis. And now there's this cool new stuff called Bayesian analysis, and these are the advantages. However, McElrath is not doing this. He's instead building, I would say, a system of science, which is amazing. Like, he just, it basically boils down to use your brain before you do anything. And it's so simple, but still amazing. Like it goes, when you go through the chapter and read it a second time, it goes into the direction of open science and everything like this. But instead of just bashing frequentist analysis, he builds an own new way of doing science. And I think this is really positive and amazes me.
Totally agree. I've been in a few Bayesian workshops so far, and usually the first lecture is always devoted to telling people that everything they've done so far, which was based in frequentist analysis, has been a steaming pile of trash and they need to do better. And that is one surefire way of turning people off of what you want them to take away from what you're doing. So this, I fully agree with you. It all comes back to me reading that chapter and to myself saying, I wish I could write as compelling as he does, because the chapter was such a breeze to read.
So actually what I would like to add is that when reading the first chapter, I also thought about my own work, of course. What I always like to do is actually to get some scrap paper and scribble, when I do work. And this is actually what he also recommends, not just opening your statistics program and doing a t-test or whatever, but scribble some relationships on paper or whatever. So I really like that he recommended that. And another thing I was thinking of is that I do work with a tree species on the Galapagos Islands. And there's only a few individuals left, so not even 1,000. And I took samples of 10 by 10 meters, and of course I don't have many samples. So should I just not work with this endemic tree species that is almost extinct because I don't have enough samples, or can I just take Bayesian statistics and actually do something with it? So that's actually an aspect I liked about it.
I get that. Sam, you look like
I don't know about you guys. When I saw that flow chart on frequentist analysis, I literally follow that every single time I do my. So I was like, I feel like I'm looking at myself in the mirror on a really bad day, and it's like you're just looking at yourself in the mirror. It's like, Jesus, I've been doing this for the past 5 years. It's like, even when you're like constructing experiments, constructing analysis, or even when someone comes along and asks for some help, it's like, oh, let's go through, let's think of how you can actually construct this in a model and do this. It's like, oh, but no one tells you this when you learn things. It's like,
That comes
my
back
reaction.
to what I often hear from my students in my statistics classes, and through no fault of their own, because I haven't taught them any better. They say, I have selected this method because the data are like this. And that very much reads like a flowchart of saying, if this, then that, which only tells them to have, it goes back to that cookbook analysis, or not analysis, but metaphor that I mentioned earlier, where people just pick stuff off a shelf and then put it back in. And even though that was one thing right in the beginning of the chapter that sounded very daunting to me, as in, especially for the Germans in the room, I feel as though we have this very big respect for engineers, and him saying, by the end of this, you're basically supposed to be engineering your own statistics, made me go, slow down. I don't know if I'm going to be comfortable with that, but I'm curious to see. So yeah, Sam, just cultural stuff here. If you want to just blow to someone in Germany, just tell them you're an engineer, because engineering exams at German universities are just horrible.
Learn something new every day. I wasn't expecting to learn about German culture in a basic.
Yeah. But Anna, you mentioned this scribbling something when you start off a project, and that very much resonates with me. Sam will be able to attest to this, because when we had our nerd out session, I just pulled up the whiteboard on Zoom. And if my supervisor ever listens to these recordings, I really like whiteboards, just as a hint. So for me, whiteboards are such a valuable asset in any kind of office space. And I've made use of them so much at my last university in Bergen, where I did my master's, because drawing flowcharts, not necessarily drawing flowcharts that lead to a certain test, but drawing flowcharts that visualize how I obtain my data, how I treat my data to get them in line with one another, so that I can then do something with them, is such a good way for me to spot potential mistakes in what I do.
That's basically a DHE, right? A deck.
Exactly. Yeah.
It's just sketching things, like
yeah,
giving your ideas to paper.
It's just, I think also the big advantage is just when you do it quick on paper, you don't have to think about technical issues. You just get it done on paper. You know, you're not limited by your laptop, whatever. So I always like this as a start. Or the whiteboard, of course, yeah.
Yeah. For a while, I've tried to just make some DAGs in PowerPoint, but then I noticed that I was just way too cumbersome for my liking. So I've just reverted back to having a big chunk of paper in my office and then just one pencil to just scribble stuff down on. But I don't know what it's like for you guys, but whenever I go not just into a research project, but also when I start coding, I usually like to start off with a flowchart of how my code should work. And that has, I mean, I'm just, Sam can attest to this, but I'm a big proponent of flowcharts. Almost all of my life is organized on flowcharts, or lists, or tables, but probably one of the three.
I just recently did this with a course of mine. We had a paper almost finished, and I think we kind of applied a flowchart method. We just printed it and cut it into pieces and just aligned it in the most logical order. So basically making a flowchart out of the paper, and it made it so much better. So.
Oh, definitely. So I've been taught to take the figures I want to put into my paper, just put them on individual slides in PowerPoint, and rearrange the slides in order until I can finally tell a good story, and then write the paper around that. And I think that's the exact
That's good. That's a good idea.
Oh, that's it. It has helped me a lot. getting that paper into submission was still a hassle, but that's a story for another time. It feels to me as though, and I don't want to cut us short here, but it feels to me as though we are running slightly out of discussion points. And I believe that's just because this was an introduction chapter and an introduction lecture where there's not much of scientific. I was close to saying scientific merit. I think there's a lot of scientific merit in here. I'm going to correct myself and say technic stuff to discuss. But one thing that stuck out to me was that just going through the first chapter, by the end of it, he gives a short summary of how the book is supposed to be structured or how he intended for it to be read. And then I had another look at the schedule that I circulated to you guys. And I noticed that we could probably improve the schedule a bit. And I'm just going to go back into screen share here to show you the schedule as it has been intended so far and some proposed changes from my end. And I would just like to know if those changes would be okay with you guys. So I'm going to share this right here. So this is the schedule as I proposed it, right? Where next week, before going into the actual holiday break for Christmas, we would just look at chapter 2 in the book and lecture 2 on YouTube. And then after the holidays, we would pick up with chapter 3, which also is covered in lecture 2 on YouTube. Now, I know that many of us have stuff to finish before the holiday break sets in, but I would like to believe that we could gain a lot more from altering these schedules slightly. And next week, covering both chapters 2 and 3 in the book and lecture 3 on YouTube. Because then, at least according to the outline by Meg Alreith at the end of chapter 1, we should be done with all of the basics of Bayesian inference and calculations, and we would have 2 weeks of holiday break to just have that settle, and we could be able to digest a bit more before in the new year we can then go into actually building models. I would be very much in favor of doing it this way because it wouldn't artificially split up the basics of Bayesian inference by a full 2 or 3 weeks. So if that's cool with you guys, I will just send around an email just saying this is the updated schedule, which also cuts down on 1 extra session, which might make people happy, just with a slightly higher workload right at the beginning here.
Sounds great.
Cool.
Well, yeah.
Yeah. So it's just the 2 chapters in the book and the 1 lecture.
Exactly.
Gotcha.
I think that's going to be manageable. And like I said earlier, if you don't have the time to prepare for these sessions, be that fully prepare with all of the stuff I suggest in the schedule, or just partially, don't worry about it. I would still be happy if people just showed up and weighed in with stuff they don't understand, even if it's stuff that in the summary of whoever's presenting, which most of the time will probably be me, if you show up and you haven't read the chapters and you haven't watched the lecture, and then in the summary I just gloss over something that doesn't make sense to you, even if that may be, so it might be because I haven't done a good job at summarizing stuff. It might also be because you couldn't prepare for it due to any number of circumstances. And that can even be just didn't feel like it. Even if that's the case, just asking a question of, I didn't get this, could you please explain that a bit again, will definitely make it much better for whoever gives the explanation to actually understand what they're talking about and prove that they do. And for everyone else on the call, it will be a really good exercise to see if they fully understand what was said, and everyone can weigh in at that point. So this all comes back to not being ashamed for when you don't grasp a certain concept. Because I believe, especially with Bayesian statistics, that happens to a lot of people because they get overwhelmed with a lot of information dumping in quick succession, especially on 1-week workshops for Bayesian analyses. And then people don't feel comfortable enough to speak up about it. And I wouldn't like us to fall into that trap.
Sounds good.
Cool.
Just another question about the sessions next year. Are you also including R scripts? I mean, can we ask questions about R things?
Yes.
I mean, I think that's going to be one of the biggest issues, I guess.
That is exactly my motivation for making these 3-hour block time slots. And I don't expect everyone to keep those 3 hours free. If someone has to leave early, if someone drops in late, all of that is totally fine. This is very much open room. Everyone comes and goes as they please. So I would assume that we will probably almost always be done in at most 90 minutes with discussing the contents of the chapter and the lecture and the summary and having just a lively discussion about it. Afterwards, the next 90 minutes, with probably a coffee break in between, I would argue we should keep open for people to come in with their own issues. So if you have a script that doesn't compile, come in, show us, we can talk about it. If you can make it into a minimal working example, that'd be even better so that people can run it simultaneously on their machines. So we can see if maybe it's a machine issue. I have run into a couple of those currently trying to run some Bayesian networks right now, which I'm still very uninformed of. So I'm having a hard time troubleshooting, especially if it works on my PC at home, but not on the server. And I have no idea what led to that. But then it might be good to just circulate to tell the people to see if maybe they can run it, and it might be a package issue in R. The book that we're going through definitely has some R examples, which we can go through. And I would actually like that. So I think starting in the next few chapters, there will be a few problems at the end of each chapter. And I will make it my goal, I can't promise I can manage that every time, to work through them. And then we can talk about them after the fact. So at the actual session, I can't promise I'll make it all the time. And sometimes I might not even be able to do it just because I don't understand it. And that's why I really want some help from all the others. Also, I am looking into the book of An Introduction to Bayesian Analyses Using Winbugs by Mark Carey. I will also upload the PDF to that. I will just quickly have to check if you can actually download it off of the internet, because if you can't, then I probably shouldn't upload it to a cloud drive, freely available to people. So I'll check that and then see if I can upload it. But that book also includes exercises, and I will also include those into the session material. So when I'm giving a summary, I will try to also make a separate presentation or put it into the same presentation for the exercises mentioned in both books that go well with the topic of that session. And then we can work through them together. And if people come prepared, having made solutions to the exercises, that's grand, especially if we end up with different solutions. Because I think then we can talk about how we arrive at that point. But yes, R scripts, to get back to the question and give you a short answer after I was rambling for so long, yes, R scripts will be part of this.
Perfect.
All right. That is actually all I wanted to get through today. So I am totally happy with letting all of you guys off the hook now after just 1 hour of a proposed 3-hour schedule here. And I just like to say thanks for showing up, and expect a update email from me later in the day. Once I've compiled the recording and uploaded it together with all of the other material, it'll all be available to you guys.
Thanks, Eric.
Thank you.
Welcome. Also, sorry, just one last thing. If there's anything you would like to see covered in here, which may not be in the book, just drop me an email, and I'll make sure we sort that out somehow. You will also see that I have already got my mind on a few people whom to invite for input talks. If there are other people who you would like to approach for some input talks on this, just do it or let me know and I'll organize it, because there are a lot of really talented Bayesian people out there who I think if we can just pinch a bit of their time off of them, we could greatly benefit in just listening to them and interacting with them.
I might have
And that's
some,
all from my
actually.
side.
Yeah.
You do? Lovely.
I'll see if they're available.
Cool. Just drop me a line about them.
Will do.
All right. Thank you very much.
Cheers,
guys.
Eric.
Thanks. Bye.
Bye.